So until now we have used edgeR or dissect2 in order to find genes that are differentially expressed in two classes. The two classes that we had was the factor was the infection, and this factor has to level the infected class and the control class. And what we have found are genes that are differentially expressed in these two categories. So these differentially expressed genes are split in two classes, in two categories: the overexpressed genes. This means that these genes are highly expressed in the infection class compared to the control class, and the underexpressed genes, these are the genes that are expressed lowly in the infection class compared to the control class. So of course this is relative to each other, which means that depending how we do the comparison, if this is first and this is second, or if this is second and this is first. And overexpressed genes means a list of genes, the names of the genes, so gene 1, gene 100, 1, and so on, that are highly expressed in the infection class compared to the control class. And this is another list. So a gene from the organism either will belong to this class or to this class, or to genes that are not differentially expressed. Okay. And the next question is to find out what these genes are doing statistically. So what are the functions, the molecular functions of these genes, where they are in the cell, these genes, and so on. And this gene ontology analysis refers to a group of genes. So it's not an analysis that is done in every gene separately, but refers to a group of genes. For example, the overexpressed genes, so this list here. Okay, and let's see how we approach this problem now, to find out what these genes are doing, or with what attributes, with what terms they are related. So for example, gene 1 might be connected, several biological terms are linked to each gene. So for example, gene 1 might be linked to the following terms: response to biotic stimuli. So this gene is activated when there is some biotic triggering here. Can be another term as well. For example, immune system process. And so on. So a gene might be linked to several terms. Another gene now, for example the gene 101, might be linked to this term as well, and then to another term, and then to another term, and so on. And another gene might be linked to other terms here. So from our list of genes, we have that each gene might be linked to different biological terms. If we would take all the genes of the organism, so gene 1, gene 2, and so on until gene 1000, I don't know, let's say 50,000, then each one of these genes are also linked to some terms, biological terms. So for example, term 1 here it might be response to biotic stimuli as this one. Maybe I should put this, since this is gene 1, I will put this gene 1 and gene 2 here. Immune, and so on. Gene 3, it will be term 1 again, let's say, and so on. And we have the whole list of terms linked to every gene in the organism. So this one here is a subset of all these genes here. Here we might have 500 genes that are overexpressed, and here we might have 50,000 genes, like here. So how we study if we have an overrepresentation of some terms here compared to this group here? Or in other words, how we study which biological terms are found here more often than what we would expect by chance. So our biological question is, let's find if these genes are doing something special. Do them overrepresented genes overexpressed do something special, or in other words, are they linked, in other words, what are they doing? Now this is not very specific, and more specifically what we want to answer is if they are linked to some biological terms more often than what we expect by chance. Okay, let's see how to answer this question. We have these two groups here. So we have our group of genes that are overexpressed. Let's call this overgroup. And we have also the total set of genes. Let's call it total. Now, assume the following, that in the overexpressed group here, this term, response to biotic stimuli, is found in total 200 times. So out of these 500 genes, 200 genes have this term here in the description. So not everyone has it, but 200 genes of these 500 have it. Now here in the total, this term, the same term, is found 1000 times. Of course, we expect to find this term more times here than here, because this set is a subset of the genes here. So out of 50,000 genes, 1000 are linked to the response to biotic stimuli. Okay, now we go to the statistical question slowly. So let's see how to formulate the statistical question. Our null hypothesis is that this set of genes is nothing special compared to this one. So it's like that we closed our eyes and we got, without seeing, 500 random genes from here. So the null hypothesis says assume that over the list over here is nothing special, which means that it's just a subset, it's just a random subset from total. And the alternative says that this is not true, that the overlist is not actually a random subset, but they are indeed genes that are doing something special, that are linked to some special biological process. It's not a random subset from all the genes. Okay. Now we want to calculate a p-value for this hypothesis. So it goes like that. So if we would assume that the null is correct, then this means the following: that it's like that we have a box here with 50,000 genes, I will draw it like that, and I will explain later why some are filled and some are empty, some are blue and some are white. I have 50,000 genes, 50,000 balls here, and there are, let's put it, yeah, the white ones are 1,000. So here the white balls represent the number of genes that are linked to the term response to biotic stimuli in the total list. And these are, since these are 1,000, we have 1,000 balls that are white here, which means that we have 49,000 balls that are black, since in total there are 50,000 balls inside here. Okay, so this is the total. What is the over group? The over group says that I have a box of 500 balls, and there are here 200 white. Okay, now if the null is correct, it means that these balls are inside here, we closed our eyes and we just randomly picked 500 balls. And then we saw them, and we saw that 200 of them are white. And we want exactly to evaluate how possible is this to happen by chance. By chance meaning that we just close our eyes and we get randomly 500 balls from here. So how possible is to find 200 or more white balls if we just randomly pick 500 balls from this group? Now, if this is possible, if this probability is not very small, so it's quite large, then we will say that these 500 genes are not anything special compared to this total list of genes. So it's really like it's a random subset of those. If this probability is small though, we will not be able to say that this is a random subset of these genes, because it's really small probability to get so many white, or in biological terms, so many genes that are linked to response to biotic stimuli. So in order to assess this probability, we use a distribution which is called hypergeometric, and it has the following parameters. The first parameter is the number of successes, or the number of white balls that we got eventually. We got 200. And the second one is the number of white balls that exist in total, which will be 1000. The third one is the number of black balls, white in total, I will write here. The third one is the number of black balls in the total. This is black in total. So this is this is in the overrepresented the white. So the white in the overrepresented. And here is how many balls we got in total in the overrepresented group. We got 500. This is the size of the overrepresented group. Now you can understand that probably this probability will be pretty small. Why? Because in our group of overrepresented genes, the proportion of white balls, or of the genes that are linked to biotic response to stimuli, response to biotic stimuli, is about 40%. So it's 2 out of 5, which is 40%. While here in the total, the proportion of white balls, or of the genes that are linked to the term response to biotic stimuli, is 1000 to 500 to 50,000, which is 1 out of 50, which is 2%. So even though the probability to find the term biological response to external stimuli, to biotic stimuli in the total group of genes is 2%, in our list of genes we find it in 40% proportion, which is huge compared to this 2%. And this probability here tries to evaluate exactly the probability to find such a huge overrepresentation of this biological term just by chance. So this is what we are doing, and what we do in practice is that we get all the list of overrepresented genes here. I just put it in this box of the tool called gProfiler. So let's do it from the beginning. Let me copy this. Just write in Google gProfiler. Go here. I paste the set of genes. I choose C. elegans because this is my organism. Elegans. Okay. I just click Run Query. So some terms here do not have a clear meaning, and we will ignore them. There are several, but doesn't really matter. And here are the results. They try to summarize all the results, but what we will do is that we will go to the detailed result, and this is what we see. So gene ontology, these terms that we said previously, are separated in three categories. One category is the molecular function, MF, another one is the biological process, BP, and the third one is the cellular component, which is the CC here, where on the cell this gene, with what cellular component this gene is linked. Besides these three terms of gene ontology, the molecular function, the biological process, and the cellular component, there are other terms that the gene might be linked to. This is a pathway from the database called KEGG. These are some other pathways. These are transcription factors that these genes might be linked to, and some other biological categories that developers of the gProfiler have added recently to the tool. Okay. Now let's go to the biological process, which is easier for us to understand. So there is indeed this term here, response biotic stimulus, that has a p-value 5.7 by 10 to the minus 44, which means that, let's see the details here. So our query, so this list of genes that we put, has a size of 372. Okay, I had to rerun the analysis in a way that will match what I told you on the paper, and then we'll change it back. So let's continue, and then we will see the corrections. So for example, for the term, where was that? The response to biotic stimulus, my query had a size of 101,000 almost genes. So this was my list, and out of this, and in the total there are 46,000 genes. So the whole C. elegans contains 46,000 genes, 47 almost, and my query 1,100 overrepresented genes. Now, in the total here, there are 142 genes linked to response to biotic stimulus. In contrast, in my class here, in my overrepresentation group, there are 63 genes linked to response to biological stimulus. So I have 63 out of 1,100, and the total here is 142 out of 47,000 almost. So there is a huge overrepresentation in this list compared to the total. this is something like it's more than 1 to 20 here, and here is something like 1 to 30 or so. So there is overrepresentation of this term in my list, and this is reflected in the small p-value here. Now the default, so here I have used as a U set, the total set, this U set, the universe, the total number of genes, and that's why you see that this number is the same everywhere. However, what's happening is that not all genes in this organism have an annotation related to molecular function or to biological process or to cellular component and so on. So it might be a subset only of these genes has an annotation term that is related to molecular function. The remaining does not have any term related to molecular function here. So the default functionality of the gProfiler here writes use only annotated genes. Now if I will do that, you will see that the numbers will change. Not so much the results, but the numbers will change. So click again here to show the results. Okay, now in this biological process thing, or maybe we'll go to the molecular function thing, which is here. Yeah, so there are only 9,000 genes that have a molecular function term. They are annotated. Now in my group, you remember I had 1,100, only 497 they have a term related to molecular function. So out of 497, I found 5 that have the term dipeptidyl, peptidase activity and so on. In the total, which is 9,876, I found 6. So again, I have an overrepresentation, and this is reflected here in the low p-value. The same is true for every term. With this way, we find out that the list of genes that we used are related to genes that have something to do with the response to biotic stimulus, response to metal ion, is immune response, immune system process, immune response here, response to external stimulus, response to stress, and so on. Remember that these are genes that are overrepresented in the infection class. So it means that the genes that their expression level is higher, is getting higher, are genes that have to do something with the defense of the C. elegans to the pathogen. Okay, now if we put in this list the lists that have lower expression values. So I delete this, I go to my terminal, I get the underexpressed genes and I paste them here. Okay, ignore those that they are ambiguous. And now results are different. So I don't have any longer response to stimuli or to stress, but I have something like fatty acid metabolism, carbon metabolism, and so on. I don't have so many results. Probably I must use I must export them in PNG or CSV. Maybe CSV will be better. just to see all the terms. Okay, and here I see, for example, fatty acid metabolism, carbon metabolism, fatty acid degradation, valine leucine, isolate leucine degradation. So it seems that something related to metabolism, which means that these individuals that are infected, they have slowed down the whole process of metabolism, maybe because of the infection, which makes sense somehow. Okay, so these are the main results from the gene ontology analysis. Again, it really does a hypothesis testing with the null hypothesis that this universe here, the total set of genes, is the same as the overrepresented, overexpressed or underexpressed or our query list of genes. It evaluates this hypothesis statistically using the hypergeometric probability distribution and the p-value, and we can assess overrepresentation of some biological terms like these ones using the hypergeometric probability distribution. Thank you very much.